Dear friends, today I'm going to speak about false memories. A little more than a decade ago, memory manipulations might have seemed like science fiction. Two scientists made a great progress in understanding the brain's work. Xu just received his philosopher degree from Baylor College of Medicine, Houston, Texas. He studied the mechanism of learning and memory. Steve Ramirez, a 24-year-old doctoral student, created with Xu an experiment which has advanced a new era in memory research. In the first study, published in Nature in March 2012, Ramirez and Liu identified, labeled, and then reactivated a small cluster of cells encoding a mouse's fear memory, in this case a memory of an environment where the mouse had received a food shock. The team focused their attention on the top of the dentine gyrus, where contextual information such as place and time is recorded. They trained mice to associate a particular room with the shock and tagged the corresponding memory trace in the dentine gyrus. They used engineered wires, which contained biological sensors, which turn on after the cell has finished working. The next step was to manipulate a specific engram to create a false memory, an Elcott experiment detailed in Ramirez and Liu's second paper, published in Science in July 2013. They put the mouse in the box without shocking it. A memory of this experience was encoded as an engram. The following day, the mouse was placed in a different box, where its memory of the first, safe box was triggered by shooting the laser into the dentine gyrus. At that exact moment, the mouse received a food shock. On the third day, the mouse was returned to the safe box and immediately froze in fear. It had never received a food shock there, but its false memory, created by the researchers in another box, caused it to behave as if it had. Ramirez and Liu have recently taken on another big memory question. Can we intervene in a depressed state in an animal by reactivating positive memories? They let the mice spend time with the mouse of the opposite sex, while labeling active trace neurons in the dentate gyrus. Then they stressed the mouse by immobilizing them. When the mouse was lifted with its tail, the mouse spent less time struggling than a non-stressed mice. However, when the team reactivated positive recollection, the mouse immediately acted like it felt better. It also spent more time struggling. To conclude, the inroads that Ramirez and Liu have made into the mechanism of memory are opening a wide new world of possibilities. Strategies to make lab animals forget and remember explain how memory works and may inspire new solutions for neurological diseases.